Hi everybody, I'm Marc Antoine Guery, second year PhD student at Antoine Claessens lab, and I would like to tell you about malaria parasite Plasmodium falciparum genetic diversity in the Gambia. The Gambia is a country where there are two different seasons. The first one is the wet season from August to November, and the second one is the dry season. Malaria in the Gambia is seasonal, which means that there is a high prevalence during the wet season, and malaria in the Gambia is also regional. There's a higher prevalence in Eastern Gambia than in Western Gambia. This study will focus only on Eastern Gambia. I would like to answer three different questions during this talk. The first one will be, do we observe genetic clustering at the spatial level? The second one, what is the lifespan of a specific strain? And the last one, is genetic diversity shaped by wet and dry seasons? Previous studies have already addressed some of these questions. However, there were some limitations. The first limitation would be the duration of the study, that could be of only 1 season or 1 year. The advantage of our study is that it lasts 2 years, so it includes 2 full cycles of wet and dry seasons. The other limitation is the sampling selection bias of only symptomatic cases. Our study focuses on field samples, which means that we did not preselect any participants based on their symptoms, so we included also asymptomatic hidden cases of malaria. The sample collection took place in the Gambia, a Western African country, especially the eastern part of the country, in 4 different villages, as shown on the map on the top right of this slide. Blood samples were collected for 2 years, and Plasmodium falciparum presence was tested by PCR. You can see on the bottom right of this slide the prevalence that we calculated over the different time points from the beginning of the study in December 2014 to the end of the study in December 2016, with the dry and wet seasons highlighted. As you can see, the prevalence is rather mild, with a value of about 15%. The positive samples were sent to Sanger Institute that provided us barcodes and whole genome sequences. You can see on the bottom right of this slide the number of sequences of samples that were barcoded in green and the number of samples that were whole genome sequenced in red. What is a barcode? A barcode is actually a selection of 101 biallelic SNPs based on their ability to describe the most diversity in the population. As you can see on this sketch, the SNPs are spread over all 14 chromosomes of Plasmodium falciparum. So basically, we had 2 data, barcode data and whole genome sequences. We first decided to improve our barcode data by using the whole genome sequences, since they obviously had the 101 SNPs. The filtered and improved barcodes, made of 84 SNPs, were then used to calculate, to estimate, and to study the genetic diversity of barcodes by a method called identity by descent. A brief word about identity by descent. Identity by descent is actually a metric of the genetic distance between pairs of sequences. It is based on the proportion of similar and different fragments of the pairs of sequences. Its values ranges from 0, completely different sequences, to 1, completely similar sequences. Thanks to all the values of IBD of all the possible combination of sequences, it is possible to infer the most likely family tree, as shown on the right on this slide. We decided to do so using the whole genome sequences, since this data contained 20,000 good quality SNPs. Using this very precise data, we could retrieve family members using the identity by descent. Here is an example of a family tree that we were able to build using IBD values obtained from the combination of 4 different strains. In this example, we hypothesized that A and B might be the origin of AB1 and AB2 by an event of recombination. Indeed, AB1 and AB2 shows an IBD of about 0.5 with both of their parents, which means that 50% of the genome is similar to one of their parents, and the other 50% is similar to the other parents. This is the most likely event to happen when a recombination event occurs. The first thing we wanted to know is how diverse is our dataset. To do so, we plotted here the value of IBD obtained from all the combinations of sequences we had in the dataset. As you can see, there's a lot of values of IBD that are lower than 0.5. Actually, 99% of the values of IBD are below 0.5. When you zoom in, you can clearly see the difference between values of IBD above and below 0.5. So our dataset is extremely diverse. To answer the first question, we decided to base our analysis on Alfred and Mwesigwa study performed in the Gambia 1 year before our study in 2013. One of the results of this study is particularly interesting for this question. I show you here the graph they provided in their study. And so it shows the percentage of similarity between parasites, and they are grouped by the distance, geographical distance between these parasites. As you can see, there's a decrease in similarity when you increase the distance between the two parasites. Then we can say that there is a strong correlation between the spatial components and the genetic diversity here. Coming back to our data, we decided to test this hypothesis. Here, we took all the different pairs of villages and we calculated the average IBDs obtained between pairs of sequences. We grouped these pairs of villages, whether the sequences came from the same village or the sequences came from different villages. Here is the plot showing this result. On the left, you have all the data points obtained from sequences coming from the same village, and on the right, the data points obtained from the sequences coming from different villages. In this situation, we could not say anything about it because we think that we don't have enough data points to infer anything. So we would need more data points, hence more villages. We did exactly the same, this time using households. In this case, on the left, you have data points showing the IBD values obtained from sequences coming from the same household, and on the right, from sequences coming from different households. As you can see, we could not spot any difference between these two groups. As a result, we can conclude that parasite coming from the same household is not more genetically related to parasites coming from the same household. Then parasites seem to ignore household barriers in our case and recombine with other parasites coming from other households. The second question was the lifespan of a specific strain. To do so, we compared all possible pairs of time points of our dataset and we calculated the average IBD obtained from sequences between all these time points. We ordered the time points by the time separating them, and we obtained the graph I show you on this slide. So here you can see that there's a steady decrease of parasite similarity with the time separating the time points. After 400 days, we could not see any more decrease of the parasite similarity. We concluded then that if you see a strain at a moment, 400 days later, you would likely not see it anymore in the population in this case. The last questions was, is there an implication of wet and dry season in parasite similarity? To do so, we decided to group the samples into two categories. The first one would be parasite collected during a dry season with parasite collected during the previous wet season. The second group would be parasite collected during the dry season with parasite collected during the following wet season. We then compared the IBD values obtained from these groups, from time points coming from these groups. And on the graph on the right of this slide, you can see the result. Indeed, the group with the green dots seems that it has a higher similarity than the group with the blue dots. This means that the dry seasons are more related to the previous wet season than to the following wet season. I show you here one possible explanation of this result. Here, we hypothesize that during the dry season, there is a lack of recombination events, and then the dry season is actually just a subset of the preceding wet season. However, the following wet season, there is a lot of recombination events, and so it brings a lot of genetic diversity. As a result, we can say that the dry season are a reservoir of parasite strain waiting from the wet season to recombine with the other strains and bring new genetic diversity. To conclude, I'd say first that there's a high diversity in our dataset, which is unexpected given the mild value of prevalence that we had. Second, using the households, we could say that people sleeping under the same roof are not at increased risk of transmitting a particular parasite strain to each other. Then, the likelihood of retrieving a specific strain 1 year, which is actually one full cycle of dry and wet season, is negligible. Dry seasons may be a reservoir of parasites waiting for the dry season to recombine. I would like to thank every people implicated in this project to make this work possible, and I would like to thank you for your attention.